So, very good afternoon to all of you. I hope I am audible and my voice is clear to everyone right now.
Yeah, it's audible.
Okay. So, myself, Dr. Sabdarali. I'm the convener for the program of Bio-Illumination, which is a lecture series that we have been started by the Department of Biological Sciences, Aliah University, with the sole purpose of having a platform for interaction between scientists from all over the world to different researchers, young researchers, as well as faculty programs. So, with this is the second day for 2020 series, and as we all know that IPL is coming next week. So, in lieu of that, we are having a double header today. That means we'll be having two lectures today back to back. So it will give us enough food for thought for the scientific minds to digest, and I hope all of us will enjoy the wonderful sessions that we are going to have. So without wasting much time, I would like to call upon our head of the department, Dr. Muddasarul, who will be chairing the sessions today, to please take over and start the program. Over to you, Dr. Alam.
Okay, thank you, Dr. Good afternoon, everyone. To the speakers, esteemed colleagues of our university and other colleges and universities, and other participants, students and scholars. I hope that all of you are doing well during this COVID-19 pandemic prevailing all over the world. From last Saturday, we have started this weekly talk series where we are inviting esteemed scientists working around the world in diversity of biological sciences and other successful academics to deliver their lectures and the research topics of their interests. As Dr. Ali said, the basic idea behind this was to enrich particularly our students with basic knowledges of those research fields and to motivate them to build their career in research and academia. But we are also fortunate enough that we are getting so many participants from other academic institutions also. And for that, I want to express my heartfelt gratitude to all of you for successful. And on the behalf of Department of Biological Sciences, I want to welcome all of you in today's lectures in this lecture series. So moving ahead, let's meet our today's esteemed speakers. We are going to have two back-to-back lectures today, one by Dr. Shubhasis Haldar from Ashoka University and other one by our colleague from our own department, Dr. Muddasarul Hoda. So let's meet Dr. Shubhasis Haldar first. Dr. Haldar is currently working as an assistant professor of biological sciences in Ashoka University, Haryana. He has done his graduation and masters in chemistry from University of Burdwan. He has completed his masters in the year 2007, and after clearing the CSIR NET for twice, he has joined his PhD in the lab of Dr. Krishnananda Chakraborty in Indian Institute of Chemical Biology, Kolkata, and completed his PhD in the year 2013. After that, he moved to the Department of Cellular Biochemistry of Max Planck Institute of Biochemistry, Germany, and worked for 2 years as postdoctoral fellow during 2013 and 15. Then he moved to University of California in 2015 and worked for a year. And again, he joined during 2016 to 18. He worked in the Department of Biological Sciences of Columbia University, New York, USA. Dr. Haldar has achieved many things in very young age, and has published more than 17 publications in very highly reputed journals. And most important of those are Nature Chemistry, which has an impact factor of 22, Nature Communications, Cell Reports, Angiochem, Trends in Biochemical Sciences are a few to just mention. He has received also a number of awards in last couple of years or so. He has received in 2019 the Ratna Phatkya Award by Indian Biophysical Society for outstanding contribution to Indian biophysics under the age of 25 years. He has been awarded the core research grant of rupees 75 lakhs in 2019 by SERB DST, Government of India. He has also received Ramalingam Re-Entry Fellowship by Department of Science and Technology, Government of India in 2018. He has also received the Inspire Faculty Award in 2015 by Department of Science and Technology, Government of India. He has also received a couple of travel awards also, one from International Biophysical Society and another from Asian Biophysics Association. So, not wasting much time, let's move ahead to hear from Dr. Haldar, who will deliver his lecture on single molecule technologies, a new window to see complex biological phenomena. So, over to you, Dr. Haldar.
Thank you, Dr. Alam, for the nice presentation, and thank you so much for inviting me. I hope everything is audible, and now I'm going for my presentation quickly. And yes, so I think right now everything is set, and you can see my screen. So
it's visible.
It's visible. Yeah. Thank you. So okay. Let's talk something new about that. So you know biology, you know how protein work, how DNA work. Now what today I will show you that you know what is single molecule techniques, why they are important, and what the new information you can get it from your experiments with the single molecule tools. So I will start with very basic so that you will not face any problem on that. So at first, when we are talking about single molecule, let's at first clear one idea. The idea is that how small the molecule is. When we talk about one DNA molecule or one protein molecule, what is the idea of the size, that how small that could be? So I took a real-time example that we all use salt in our houses. So of course you know that how big a size of a small salt grain is, right? You see that this is very small. So now if you divided that salt grain into 10,000 times, that would be a size of a cell. And now if you basically divided that cell into 10,000 times or 20,000 times, that becomes a size of a small protein. So you can understand that this protein, the DNA, they are really tiny, really small. So to understand them that how they are working, you need special equipment to detect that. And today I will tell about our recent discoveries of one of the single molecule techniques, and I will tell you that how important that could be to understand the biological phenomena. So when I'm talking about the cell is a complex phenomenon, right? Like a lot of things is happening in cell, and interestingly, in one cell you can think about there is 10,000 different proteins. The important thing is that, interesting thing is that they are always working for you. Even if when you are sleeping, these proteins are working for you. And now the issue is that, now the problem is that if the protein can work fine, you are working fine. But if the protein is not working fine, there is a problem. If they are not folding properly, you could have a problem with the Alzheimer. You could have a problem with the cancer. So assume that right now we have 55 people involved in the talk, and unfortunately after the age of 60, we might find out that 1/8 of the whole population of the whole 55 people might affected in Alzheimer. Or if we think about the cancer, then we don't need to wait for the age of 60. Cancer could happen to anyone in any time, and all are actually directly related to how the proteins are behaving. So let's see a small video to see that how this network is working. So you see that protein folding is going like crazy. They happen all the time, and they happen when you are sleeping. They are still working. yes, they are fine when they are folding rightly. But as I mentioned, that when they cannot fold perfectly, you have different diseases. You have Alzheimer's, you have cancer, and several others. Right? So now let's talk about one thing is that, okay, we learned that they're tiny, we learned that their folding is very important, but the point is that, what is the way out to monitor such a complex system? And there is not only one protein, there is billions of the protein working together in the cell. So what could be our way out? So, I thought, okay, Dr. Zafar Ali is talking about IPL, I thought, okay, so let's talk about football. So, what we are doing in a soccer field. So assume that we know there is a particular protein. The protein name is anything, that could be anything, that could be tau, that could be alpha-synuclein, anything. And I want to know exactly the protein is moving. So what the way I could see that? So assume that you went to see a football game, and then you are interested in only a single player. So how you will monitor that particular player? So okay, let's see a game, and let's see that how we can exactly monitor how the player is playing, how the player is moving his right leg, how the player is moving his left hand, right hand, all these things. I want to monitor a particular player. So how you can do that? So let's check this video. So you see that, this is one of my favorite players, Messi. So, you see that how he is dribbling, how he is moving his body parts to basically get past one by another. So in the real time, we also wanted to know that, how the individual proteins are behaving. So to do that, you can think about an idea that how we can do that. So for me, the easiest part would be that if I put a red ball, not a red ball, red bulb, which flashlight on his left leg, a yellow bulb on his right leg, and then, green one on his left hand. So the whole idea is that when he moves his left leg, the yellow light flash. When he moves his right leg, the red light flash. And when he moves his hand, the green light flash. So if you are thinking that I'm only talking about soccer, no, it's not. I'm talking about science, and this is the exact way, how the single molecule real-time sequencing is working. So here you can see that. So here, all these tags are actually labeled with different dyes, and when the green light flash, you can see the green light. When the yellow light flash, you see that something different is coming. And this is how this is a general way to see that how the single molecule fluorescence is working. But now there is something else. So so when we are talking about single molecule are based on force spectroscopy. So these techniques are actually based on force. So now if we are thinking that why force is important, from the slide you can already have an idea that why force is important for our body. If you still don't understand, just keep the hand on your left chest, you can find some vibration. So force is playing some role. Of course, when you are coughing or when the blood cell is flowing, force is playing some important role. So people are trying to develop these single molecule techniques based on the force. So I spoke about some very rough or crude idea on that. You can understand that what the force is doing in your body. Now let's talk about the molecular level. So if you talk about the molecular level, you see that force is also doing some very interesting thing. When the cell division occurs, or when the cell migrates, or when the model proteins, they basically carry some force one end to the other end, all the time force is playing an important role, and we basically measure that the force is around 1 to 1000 piconewton. So this is a large range. So now you can tell me that, okay, so force is important, single molecule is important, but is it not that the techniques is always already available there? So I will tell you, yes, techniques are already available on force. We have two important techniques available on force spectroscopy. One is AFM, which you might heard of, as well as the optical tweezer. So there is some limitation on both the techniques. The limitation is that when you are applying force with the AFM, you can only apply higher forces, like more than 50 piconewton. So lower forces, you cannot understand it because there is a high thermal drift. Very similarly, when you are using optical tweezer, you are talking about low force, less than 15 piconewton. You cannot apply more force with the optical tweezer because your protein gets detached. And the second thing is that with the AFM, you can monitor maximum 50 seconds. With the optical tweezer, maximum 100 seconds. So we actually developed one technique, we called it covalent magnetic tweezer. And the advantage of this technique is that it can monitor high force as well as low force. So it can monitor the all force range. And the second thing is that with the AFM, you can monitor maximum for 50 seconds. With the optical tweezer, you can monitor for 100 seconds. With the magnetic tweezer, you can monitor a particular single protein for constantly 1 month. Right? So there is a huge technical advancement, and so we will basically see that how basically we use the advantage to understand complex biological phenomena. So I just wanted to show you that this is the instrument I am talking about. So instrument is very easy. You have a magnet, and just below the magnet you have the samples here. So how you control the force? You basically change the magnet position, right? And in the sample, there is one protein. I guess this is here. This protein is actually attached to the glass. So it's very easy. So this is the place. This is the surface. This is the protein attached to it, right? And at the top of the protein, there is a small magnet. So what happens is that when you are changing the magnet position here to here, it actually feels the force at the top. So as you are applying force on the top, it actually faces, the whole protein faces exactly the same force as you are changing the magnet position, right? So okay, I'm explaining it once again because in future might be you have some problem. So this is the surface, this is the whole protein. At the top there is a small magnet, and you have a permanent magnet here. You are basically changing the distance, and as you are changing the distance, you know that magnet is actually applying more or less force. So yes, so this is a live image of how we are doing the experiment. The black one where my cursor is, this is actually the permanent magnet. This is here, my sample chamber is there, and the sample chamber is liquid. So you can do it with the water. If you want, you can do it with the cell, whatever you want. And very simply, you can understand that when you are applying force, as you are decreasing the magnet position, the force increases. When you pull it up, the force decreases. So this is the basic principles on which the magnetic tweezer this is basically built. Okay, so right now I actually also did a small trick. Small trick is something like that, where you can see the protein. The protein, what I'm talking about, the glass to the over the surface. This is not a single protein. What I did, I basically copy paste of the same protein with the octamer. That means I took a single protein, then make a copy paste it and put it 8 times. The reason is that, okay, I will tell you the reason later, but the interesting thing is that you can see 8 proteins constantly, not a single protein, right? So now I will tell you a data. That data is super complex. So, you have to go through the data very easily. So at first you see the black one. You don't need to see anything else. So if you see the black line, what happens with that? At first the force is very low. So assume this is my protein, this is my magnet. At first the magnet is far apart. So the force is only 3 piconewton. Then I'm putting the magnet down. As I'm putting the magnet down, the force increases. So the force increases up to 49 piconewton. Then we again pull the magnet up. The force becomes 6 piconewton. And then I basically put the magnet down. The force becomes 49 piconewton. Right? A simple way. So now the point is that what happens when I put the force 49 piconewton? Let's see the video here. So when I apply 49 piconewton, you see that all the domains are folding one by one. And as I have the 8 domains, I should see the 8 steps. Now you can ask me one question that, why all are 8 are not opening in a single stand. Okay, let's also talk about this later. I will not talk about right now, but you could think about during the talk why all the proteins are coming out one by one. So you can actually see 8 steps because you have 8 domains, 8 proteins, and eventually when we decrease the force to 6 piconewton, you can see that all the proteins becomes folded, and the folded one you can see there in the second step, where all the folding happens by one, right? So here 8 domain unfolded, here 8 domain folded, and now you keep the force at constant, so the protein will stay at equilibrium. Okay, so now you can ask me that, okay, what is the, why we want to do all these things? You know, what is the meaning of all these things? So, so if someone is interested about protein, if he is interested about unfolding kinetics or refolding kinetics or equilibrium, you can get all the information in a single trajectory with a single experiment. Now, let's see that what is the biological meaning of the data. So what we did, we basically choose a protein which is going outside in the cell, and then we put one protein to interact with it, and the name of the protein is DSBA, which stays in the periplasmic site. And now the question is that what is DSBA? DSBA is another protein stay in the periplasmic site, and it is introduced the disulfide bond to the protein. We want to know that, you know, whether the DSBA can also actively fold the protein and help in the translocation process. So what we did, we basically did the same experiment. Okay, this is again a confusing experiment. We at first we have 4 piconewton. We increases to 50. What we see? We'll see 8 steps. 1, 2, 3, 4, 5, 6, 7, 8. All the steps you can see that this is happening in the unfolding. Then we decrease the force to 9 piconewton. 9 piconewton is a high force, so it is not allowing the protein to fold properly. So you see only one domain folded out of the 8 domain. So the folding is much decreased. Then we basically added DSBA here, and when we add DSBA, we see the 6 domain folded. That means here only one domain folded, here 6 domain folded at the same force, which actually tells us that now if you think the same experiment in a different way, you can think about it another perspective, a physics perspective. The perspective is very simple. You are applying the force from the top end, right? You see the force, the arrow is on that side, on the above side, but the protein folding happening on the opposite side. That means the folding of the protein, excuse me, the folding of the protein against the force is creating a mechanical work, right? It is simple. If I basically pull your hand from my end, and if you actually pull the hand on your end, that means you can think about that you are doing a mechanical work, right? Very simple. If I pull the hand on this end, and if you are pulling the hand on this end, then this hand is doing some extra mechanical work. So that is the protein during folding is doing some mechanical work, and which is actually increased in presence of DSBA. So we try. So again, we can exactly calculate that by doing that exactly how much mechanical work DSBA is generating. So now if we think about the translocation process, this exactly happened in the translocation process. So this is the cytoplasmic site. The protein is going to the periplasmic site through a motile protein called sect-A. But now the point is that as the protein went to the periplasmic site, DSBA grab it, they fold it, and the fold again the force at this range actually stop the backsliding of the protein, because you know this is a narrow pore. When the protein goes outside here, DSBA fold it here so that it cannot go back, right? So thus we basically shows that DSBA prevents backsliding, and this is how DSBA actually help in the protein translocation pore. So then I would also like to show you that how the fluorescence single molecule techniques could be useful. So I guess in the textbook you all read that the chaperone is actually helping in the protein folding. So I would like to tell you that, you know, the real experiment which actually comes to the textbook saying describing the chaperoning mechanism. So, you know, we have several chaperones. So let's go back and think about the history of chaperone folding, how it starts. So history of chaperone folding starts when Kendu and Peru, they basically solved the first crystal structure. Then Anfinsen shows that protein can fold by themselves. But here, these people come in 1990 when Ulrich and Horowitz says that all the protein cannot fold by themselves, but they would need the help of the other protein, and they call this protein molecular chaperoning. I'm quick in this place because you all know the history that how chaperone works. So I'm working with a chaperone called GrowEL. So this is a molecular chaperone which has actually interesting because it interacts or it folds almost 248 different protein. Some protein cannot fold at all in absence of GrowEL, and some protein needs GrowEL to fold at the higher temperature. So let's see a small video on see that how GrowEL works. So let's see the video. So this is the ribosome. So from ribosome, this is how GrowEL works. So I would like to tell you that to explain the same thing, I would like to describe it structurally. So if you see the structure of GrowEL, it actually two rings. They are stacked back to back each other. Each ring contains with 7 units. You can tell it 7 domains, and all the domains are exactly identical. So in the same way, both the rings are identical. So at the top of the ring, there is a cage, which you see that which is coming from outside. We called it GrowES, and this GrowES makes the chaperoning system. So what happens there, for the easy describing, sometimes I draw like that. So what happens is that this is the GrowEL, right? So when a misfolded protein comes, you see that at first it starts its apical domain. So head domain in scientifically we called it apical domain. So apical domain, when the protein comes, it basically unfolded, right? Force unfolding happens. And then the GrowES comes and they basically push the protein inside. Inside the protein folded, and when the protein folded completely, it's basically go out. And in the same time, the another protein is basically comes from the backside. So this is how the GrowEL chaperone works. Now there is a huge controversy on how the GrowEL works. So one group of scientists, they believe that GrowEL, without GrowEL, the transient aggregate form, and with the GrowEL there is no aggregation. We try to understand that is it the only reason of GrowEL work, or GrowEL have some inherent activity as well. So to do that, we actually need a system, the system at which the protein doesn't aggregate. Because then you can explain that, okay, this is the condition, this condition protein doesn't aggregate, and if GrowEL is still working, then we can say that, okay, GrowEL has some additional activity. So we would like to explain that. So what we did, we used very low concentration, and we call it single molecular condition, so that the molecules are far apart, that there is not enough binding capacity of those molecules. So to understand that, we actually used another single molecule tools, and we called it the FCS, the fluorescence correlation spectroscopy. So I would like to use one slide to explain you how the spectroscopy behaves, and I'm considering that you know the background of how the fluorescence spectroscopy works. So so if you see the first slide, so in fluorescence spectroscopy we are using a lot of sample, right? We are applying light here, and we are detecting in this place. So there is a lot of light. You can see the whole window is actually red. So the a lot of lights is there. With the FCS, we are doing two interesting change. One is that we are decreasing the protein concentration to picomolar, and the second thing is that the observation volume is now on femtoliter. So what's the result is that now your number of molecules is much less. So when the fluorophore light enters, you can see the fluctuations, and when they are outside, you cannot see the fluctuations. So it's something like that. So now you are not seeing a constant light, but you see a fluctuations of light, right? So if you understand the fluctuation, that actually tells us the diffusion time, how long the protein needs to pass through the observation volume. In a simple way, this also tells you the number of particles, that how many numbers of particles exactly there in the observation volume. So that means is that if you think that if at first the number of particles becomes 4, and in the meantime it becomes true, then you can think about that there is an aggregation going on, and the aggregation is a dimer. So when we did the whole experiment, we found out that number of particles doesn't change with the time. That means that, GOEL, that means the protein is actually not aggregating at all in this condition. So, but there is an issue. The issue is that when we are talking about that at the single molecule level, which is 100 picomolar, the aggregation is not here. The question is that is the GOEL is working at the condition or not? Because if you are comparing it with the cellular level, then you can see that the percentage of GOEL is much less. So we need to now prove that at the 100 picomolar concentration, the protein is not aggregating. We need to prove that in this condition, GOEL is also working. So for that, we need to use another single molecule techniques, which we talk at single molecule FLET. So single molecule FLET, what you do is that you are actually taking the whole protein, you put a light bulb here, which is basically a tag. This is a tag, and you put another tag here. So the whole thing is very simple, is that when they are come closer, you have a high FLET. When they are far apart, you have a low FLET. Right? This is simple to understand. So, ideally you should see this trace where my slide is, where my mouse is now. You can see that at when they are far apart, the FLET is very low. You see the value is very low. When they are closer, the FLET is very high. So anyways, if you are not interested a lot on the technique, let's talk about the result. What we found out that when we don't have the GOEL, the protein is folding, but it takes almost 160 minutes. But when you add the GOEL, the whole process is finishing in 45 minutes. You can see the same state, right? Here is 45 minutes, the state you are finding in 9 minutes. Whatever you are finding in 160 seconds, you can find it here 45 seconds. So that means that GOEL is still accelerating the folding rate of the particular protein. So until then, at the first slide, we see that the protein is not aggregating. The second slide, now we see that although we are using 100 picomolar, the GOEL is still active. So now we need to understand that, okay, so this is not the reason is that GOEL is aggregating the prevention, because now we have a system where there is no aggregation, but GOEL is active. That means there's something else is happening. To understand that, we thought, okay, let's try one thing is that, let's try the chain dynamics, how the chain is fluctuating inside and outside the GOEL ring. The reason is very simple. The reason is that if it is a confined place, and if your protein behaves like a polymer, if you put the protein inside, their chain dynamics must be restricted. But the issue is that at that time, there is not technology available who can monitor the chain dynamics, very fast chain dynamics in that time scale. So what we did, we basically developed another technique which we called photoinduced electron transfer with diffusion coefficient. What we did is that we basically used the same protein with a quencher. So the point is that when the quencher and the tryptophan come closer, you can see a dark state. When they are far apart, you can see a bright state. So there is a fluctuation between the bright state and the dark state. So when you put the fluctuation, your data looks something like that. If you are not interested in that the technology on that level, I can tell you that these fluctuations actually can tell you how much the protein is fluctuating. So now if I basically see the fluctuations, I can see one thing, that chain dynamics outside the GOEL is fluctuating a lot, whereas when the protein is inside the GOEL cage, the fluctuation is much decreased. And as the fluctuation decreased, you can see that this is entropically restricted, and this entropically restricted actually allow the protein to fold in a faster mode. So again, so what we see is that when you are using this fluctuating protein, their fluctuations is much restricted in the GOEL. But if you mutate the GOEL ring with the positively charged residue, then you see that the fluctuations is not there. So we found that the GOEL reduces the chain entropy by reducing the entropic barrier, which in turn accelerates the folding rate of the proteins. So in the whole slide, I'm actually trying to show you that how the single molecule techniques could be useful to understand the important biological problems. So finishing my talk here, I would like to acknowledge a few people. One is that Professor Ulrich Hartl, he discovered the molecular chaperoning. So I worked with him in the Max Planck Institute. I work with Julio in the Columbia University. This is my group in Ashoka University, Chorobit, Soham, and Deep. And this is some of the undergrad students whom I'm working with. And thank you so much for your involvement. Last but not the least, this is our campus. So we are far apart, but I would like to show you my campus. This is my campus. We have a big swimming pool, gym center, and this is our dining community room. And yeah, this is our playing place. And yeah, that's it. So if you have any question, please let me know with this email address. And now, I'm welcoming you if you have any questions.
Okay, thank you very much, Dr. Hoda, for talk and a complicated topic, but you have elaborated or described it very lucidly through several simple diagrams and cartoons. So before going to the question and answer session, just I want to ask one question. I want to start with the question session. So actually, in the bacterial system or prokaryotic system, you have worked with magnetic tweezer with some proteins, DSBA. So DSBA, I think basically it is a sulfide group donor. It actually brings the SH group to the protein to be folded in the periplasmic space. So and some other DSB proteins are also there, such as DSBG or DSBE. That also plays some role in the folding processes. So is it just simply giving one protein DSBA with your protein of interest works in that way, or the mixture of proteins or mixture of total DSB complex is given there?
That's a fantastic question, Dr. Arun. Yeah, that's actually a great question. So we started with the DSBA protein, and the whole set of protein as he's mentioning. So I'm using a protein called DSBA. So what it works, it actually introduces the disulfide bond into the protein. So, and this is whole working as a complex, because this DSBA also get oxidized by a protein called DSPB, and you have also DSBG and DSBC. So we work with the DSBA, and the whole point of working with DSBA is that DSBA is the protein staying in the periplasm, and it introduces the disulfide bond. So now what we did, the protein we took doesn't have a disulfide bond. So the whole reason of doing is that, is it only a disulfide bonded donor, or it has some additional activity?
Actually, you are just giving the reduced form of DSBA with the protein of interest, so that SH group can be transferred to the
yeah, but the protein which I took, I actually took two kinds of protein with the same result. One protein has the disulfide bond cysteine. The other protein doesn't have cysteine at all. The result I was showing doesn't have cysteine at all. So that also helping the folding. So DSBA also helping the protein of that one which doesn't have cysteine or disulfide bond. That means that this is not only introduce the disulfide bond, this has an additional mechanical chaperone activity, and we explain the mechanical chaperone activity in terms of pulling the protein from the cytoplasm to the periplasmic area.
Okay, thank you very much. And I want to just go to Dr. Ali to just start the question and answer.
Sure.
For this talk.
Okay. Thank you, Dr. Aliah, and thank you, Dr. Shubhasis, as well, for a very wonderful talk. And I think you just took the concept of Ekla Chalo to a new level as to how a single molecule can be studied and having so many aspects over there. We have got a couple of questions. I'll take them up one by one. The first question is, what is the possible role for Broil in protecting protein misfolding diseases? And are those diseases somehow related to inefficiency of Broil? Dr. Shubhasis.
Yes. So Goyal is actually a very important chaperone, which actually helps the protein not only to stop the misfolded one, but it also rescues the protein from going to the aggregation state. So, but you also need to understand Goyal is not only the single chaperone. So at first, when the protein is coming out of the ribosome, it interacts with a chaperone called trigger factor. Then if it cannot hold, it actually push the protein towards the another chaperone system, DNA K, DNA J, and if it cannot hold, it comes to Goyal. Interestingly, Goyal also hold 10% of the protein. So 10% of the holding or misfolded protein, Goyal is responsible. For 90% of the other protein, other chaperones are there to work for.
Okay. There is another question, which is, I think it's more of a inquisition rather than a direct question about your talk. So, Serena, she wants to know that is there any mechanism or possibility about, using some single cell or single molecule techniques for cancer therapeutics or cure or something like that?
Yeah, that's also a very beautiful question. Okay. Dr. Ali, can you please mute your microphone?
Yeah, sure.
Yes, fantastic. Thank you. So, we actually explain the single molecule technique as a beautiful drug screening technology for the future. So here, assume that, so I was showing you a protein with all the 8 steps, right? Assume that the protein is P53, which is important for a cancer therapy. And now I was showing the addition of DSBA, if you remember the graph, and assume that you are testing it with a drug. So you immediately see that whether your drug is interacting with the P53 or not. So in this way, you just need to put your drug, see that whether it is interacting or not. If it is interacting, it's a candidate. If it is not, it's not a candidate. So in this way, you can actually quickly screen many drugs at a small time, and then eventually you can go to a cellular level so that you can actually save a lot of your time. So yes, it is much important. And the other aspect of this is that, if you know about the protein called taline and integrin, all the protein, they basically present in the cell surface, and they basically decided whether the cell will be adhere to which position. And, they basically sense the cell with the physical environment outside and send a mechanical signal towards the nucleus. And that mechanical signal decides whether the cell will adhere here or will pass through. And interestingly, in all the cancer metastasis, integrin mutation or taline mutation is a common part. So how they are sending the mechanical signal, what happens during the mutation is always very fundamental interest of the cancer biology. So yeah, this is a good way of thinking it.
Thank you, Dr. Shubhasis. There is another question by Mr. Asif Ahmed. What he's asking is that it's again an extension of what you just told about involvement of Goyal in different diseases, and so he's asking: are any specific diseases that is known to be caused by Goyal, or you have potentially at least involved in some diseases? So you have any idea about some specific diseases for that?
So right now I cannot think about Goyal, but I can think about only very close one, which is HSP90. So Goyal is HSP60 because it has 60 molecular weight, 60 kD molecular weight. So I can think about HSP19, which is directly again associated with the cancer, which is the last slides. So HSP90 is a highly relevant with the cancer metastasis.
Okay, so another question, and I think this is the need of the day. No seminar can have without a question or discussion about COVID. So we have a question regarding that. So can this single molecule technique be in any way applied to COVID-19 therapeutics? It's more of as a possibility rather than a question. But your thoughts on it, Dr. Haldar?
Yeah. Yes. Okay. So of course, so we all are thinking about that, and we are also in the same group who are thinking on the same line. So yes, so if you ask me that, how it basically helped to design the COVID-19 vaccine or something like that, I don't have the answer here. But what you can do is that you can really understand some basic mechanism, the fundamental mechanism on how the spike protein works. So, okay, so when we are talking about COVID, the main responsible protein is the spike protein. And if you know that spike protein has a small domain which is called the RBD domain, which interacts with the human AC2 protein, right, which cause all these things. And eventually, you may heard that in India, all over the world, there is a lot of mutation. You frequently heard that, right? A lot of mutations. So when we basically see the map of the muted patient of the spike protein, interestingly, we found out there is no mutation happened in the RBD domain, but the mutations happens outside it. And if you see that the North India, Eastern India, Western India, and Southern India, all the part have different kind of a structure, like kind of a mutation. So you can all study that, if you are applying force, that how the mutants or how the proteins are behaving are of the fundamental interest of the whole project.
Okay, so with that, we have come to an end of the questions that have been asked. And once again, I think on behalf of everyone, I think I'd like to thank Dr. Shubhasis for presenting a very complex topic in a very simple manner. And I would now request Dr. Alam to please continue with the proceedings. Over to you, Dr. Alam.
Okay, thank you. Thank you very much, Shubhasis Da. Actually, lastly, I am calling you Shubhasis Da. Actually, we were hostel mates in the year 2007. Dr. Shubhasis Haldar was 1 year senior to me during my MSc days. So thank you very much, Dada, for giving some time. You stay with us. We'll be having our next talk. So you will stay with us. Moving away, we'll have our second talk, which will be delivered by Dr. Muddasarul Hoda, who is a colleague of ours and working as an assistant professor in our department of biosciences in Aliah University. So let's meet him. He has completed his PhD from Department of Biochemistry and Molecular Biology, Pondicherry University, under the guidance of Dr. Rukkumani Rajagopalan, and he has also worked as a postdoctoral fellow at the Department of Biotechnology of IIT Madras under the guidance of Professor Mukesh Doble before joining Aliah University. He is currently working as he has more than 13 international research publications to his credit with an H index of 5. He has also co-authored very recently, he has co-authored a research reference book entitled Role of Phenolic Compounds in Diabetes Management, which was published by Springer Nature. His area of research includes application of nanotechnology and phytochemistry in drug discovery and development. His thrust area includes delineation of molecular mechanism of action of phytochemicals in molecular medicine, in addition to modulation of drug design in delivery using computational and nanotechnology tools, respectively. Moving ahead to Dr. Hoda, you may proceed with your presentation.
Thank you, Dr. Masroor. I'll just put my slide. Let me know if it's visible.
Yeah, before Masroor starts, so I just want to thank the university and especially thanks to Mansoor for arranging the talk. I had a great experience here, and hopefully everything will be finishing, and I would love to visit your university once. Thank you so much.
Sure. We'll invite you once things get normalized. We'll visit our university, and we'll be having a direct face-to-face talk from you.
Hello.
Yeah, you can go ahead.
Okay. Am I audible clearly, right? No echo?
Yeah. Audible clearly.
Okay, thank you. So the next topic is applied phytochemistry as an interdisciplinary research concept. So essentially, we'll try to revisit the concept of phytochemistry and its application in various domains of research. And it's more like trying to regain its old lost glory, that its significance in our day-to-day research life. I'm unable to change my slide.
Excuse me, Dr. Hoda, can you just switch on your video?
Yeah, 1 second. 1 second. No, actually, I have a bandwidth problem, it seems. So let me.
Okay, no problem. Go ahead.
1 second. Let me just check that again if I can try that. Yeah. So I think I'm now visible.
Yeah.
All right. So.
If there is
Let's
any problem,
begin.
we can go ahead with just switching up your video.
Sure, no problem. All right, so let's start. So the topic is applied phytochemistry and its concept as an interdisciplinary research area. So essentially, what is phytochemistry? It essentially deals with the three different domains of subject, that is, that includes plant taxonomy, plant metabolism, and organic chemistry. So plant taxonomy essentially deals with classification of
Sir,
plants.
please present your slides. It's not visible.
Is it? Okay, 1 second. Let me share that again. All right, yeah. Fine. Is that okay now?
Yes.
Okay, so yeah, we are talking about phytochemistry as an interdisciplinary subject. So essentially, it deals with plant taxonomy, which essentially is a classification of plants based on its anatomical and morphological features. Then after it deals with plant metabolism. Plant metabolism essentially is divided into primary metabolism and secondary metabolism. Primary metabolism in the first beginning of the 20th century, it was one of the sought after research domains where we discovered Krebs cycle, important pathways like Krebs cycle, Kelvin cycle, glycolysis, so on and so forth. Next, in the 21st century, it became quite obvious that the new domain of research could move on to the secondary metabolism. Secondary metabolism essentially deals with secondary metabolites, which essentially are byproducts of primary metabolism and not really essential for survival of the plants. However, they do have their own importance as a defense mechanism, or imparting color to the plant, or even imparting aroma, texture, or so on and so forth. So secondary metabolites are the ones we actually would be focusing in this particular slide. And thereafter, organic chemistry also becomes essential because it helps us in categorization of different phytochemicals, such as alkaloids, terpenoids, and phenolic compounds, flavonoids, so on and so forth. So basically, it helps us in identification and classification of phytochemicals. So these are the three major domains of phytochemistry. And in addition, since the phytochemistry has always been explored by various scientists for the benefit of mankind, so it has been explored by chemists, it has been explored by physicians, it has been explored by biologists. So it may be considered as one of the oldest interdisciplinary subjects. So why phytochemical research is really that important? So we know that there are a number of endemic plants that are yet to be explored and documented. So phytochemical research would actually help us in identification and preservation of these endemic plants. We know that we have two hotspots in India, one in the Western Ghats, another in the Himalayan belt. So these two are the hotspots of the flora and the fauna of the world. So, and almost 80% of the plant species are yet to be documented in these areas. So, in that aspect, phytochemical research could be quite handy in really documenting them and a recognition of some important phytochemicals which are yet to be discovered. Thereafter, it also helps in recognition of prevention of adulterations in natural products. We all know that natural products, all kinds of natural products are available in the market. So, if at all we could, if at a government level, we could push for certain level of certification of purity. In that case, this phytochemical research would actually help us in identification of any kind of adulteration present in any kind of natural products. For example, let's say it's curcumin, right? So we have turmeric. The bioactive compound of turmeric is curcumin, and in case if there is any kind of mixture of adulterants, we can easily actually go ahead with phytochemical analysis and find out whether there is actually any adulteration or not. So basically, phytochemical research would help us in identification of purity of natural products. Thereafter, one of the most heated topic in today's time is the climate change, and there are quite long discussion, debate going on whether the climate change is real or it's just a hoax. So, from my perspective, I personally think phytochemical research would be quite handy in actually helping us prove whether we have any scientific backup about climate change or not. From my perspective, if there is any climate change happening, they would definitely impact the vegetation of that area, and the flora of that area would basically experience a significant impact on the biochemical behavior. So, despite obvious, they would have a modulation in the phytochemical fingerprint, right? So that way, if there is any kind of climate change, let's say unnatural drought or extreme drought or too much of rainfall, that would actually may impact the phytochemical composition of the flora of that particular area, and that basically could actually help us in proving that climate change actually has impacted the region of that particular region. It has impacted the vegetation of that particular region. Fourth, and one of the most sought after applications of phytochemical research is the identification of a number of therapeutic and nutraceutical properties of various plant products. And this has been happening since ancient times. We all know that before the advent of the modern system of medicine, we had the traditional system of medicine, whose the core source of chemicals would used to be plants. So we all know that number of drugs today, even today, we have almost 60% of the drugs have at some point or the other have been inspired from the plant sources. One of the examples we'll be talking one of our final slides of metformin. We'll be talking about the evolution of metformin, where we discuss how phytochemical research has evolved and helped us in design new drugs. Then after, we also have novel small molecules, isolation of novel molecules, which could be which may not yet have been discovered. We could come up with new molecules, identification of new molecules, which could have n number of applications in diverse fields, and that again could go for patent and so on and so forth. Thereafter, one of the final and the most important aspects of phytochemical research is understanding metabolic pathways of secondary metabolites. As I said, the 20th century was based on delineation of metabolic pathways of the primary metabolism, the primary metabolites. This century could lead the pathway for delineation of metabolic path. We already are into the process. We'll deep into it, and maybe someday we come up with new molecular pathways where we can actually modulate those pathways to streamline and get optimum yield of our desired secondary metabolites from the plants. So these are the certain reasons why phytochemical research is quite important, quite significant. So the basic stages of phytochemical research essentially includes, can be divided broadly into 4 stages. Number 1 stage is the identification of the plant resource, which needs a little bit knowledge of plant taxonomy. Quite not necessary that you need to have a background of botany. You can always start learning at any point of time whenever you want to research. Even a normal layman who hasn't really gone to a formal education, gone for a formal education, even he has that knowledge of taxonomical features from his traditional information that he learned from his peers. So, subject-wise, keeping subject apart, it's all about the interest. If you even if you don't have the knowledge of plant taxonomy, you can always acquire one and really, give a formal face to that. So, identification of plant is very important. Thereafter comes the extraction process. Extraction process is quite common. We know that in our day-to-day activities, we already we perform this extraction process at home as well. For example, if we all agree, right, that even right now we are having COVID going on, and we are trying a number of homemade remedies, right, just for the sake of prevention. Just we know that okay, you have cough cold, you got to have certain kind of home remedies. So we use certain kind of extracts we prepare at home. We boil, take some spices, and boil in water, and take out that extract and then drink it. Right. So that is basically what you call extraction process, and it has a whole set of research into it that we'll be discussing in the coming slides. Thereafter, once extraction takes place, you have to remember that extract contains a number of phytochemicals. A single extract may contain hundreds of different phytochemicals. Some of them may have a bioactivity, some may not have bioactivity. Some may be relevant, some may not be relevant. However, be the case, it's always important and good to have a concentration of a bioactive compound if you have really identified any of those. You can go ahead with isolation and identification of phytochemicals. Isolation, in that case, if you have identified, and suppose even if you have isolated a new compound, you can always go for identification. And once you have identified the compound or even isolated, you can go ahead with different application stages, different applications in the stage 4. Now, the point here is you do not never need to start from stage 1 and go on to stage 4. We can always go ahead from stage 4 to again go back to stage 1, because stage 4 essentially, if we know the compound, right, or we can go ahead with the applications. And if you don't know the compound, all right, so we are into certain applications. For example, let's say I'm into a textile, right? So I need certain endemic colors, exotic color or exotic flavor. For that, I need to explore different ventures, different options, like different plants which has never been tested or tried before. So I would try to look for different plants and identify them and try to find some chemicals within them which could give me a distinct aroma or a flavor. That goes for culinary, sorry. But same is the case with textile. Exotic colors and can be generated. And apart from that, you can also go ahead with the other applications. And most of the important applications is in the field of agriculture and medicine that we'll be talking in the next slides. So as I said, identification of plant source is actually very important. And some of the different parts of the plant, almost every other part of the plant actually contributes in generation of phytochemical, extraction of phytochemicals. Among them, the roots, leaves, and the bark are some of the most sought after plant parts, which contains maximum amount of phytochemicals. Different plants have their own bioactive phytochemical, the recognized ones, and the ones which are not even recognized, that still opens up doors for investigation for us. So almost, as I said, 20% of the plants has been actually recognized and studied till date, and 80% still is left out. An entire ocean is there to explore for us. So one of the most important isolations of phytochemical research in the 20th century was digoxin. We all are familiar with digoxin. It has been isolated from Digitalis purpurea. It's a weed, and the leaves actually contain this digoxin. Digoxin essentially is a cardiovascular drug, which deals with cardiovascular diseases. It minimizes arrhythmic heartbeat. And secondly, another one is the quinine. which is taken from the cinchona plant, cinchona tree, and it has been extracted from the bark of the cinchona tree. It is an anti-malarial. Then again, we have the morphine. Morphine has been extracted from certain flower parts, from the poppy plant, Papavus somniferum. So these are certain miracle drugs which have been isolated in the 20th century. Now, in the 20th century, still awaits us to explore many such potential phytochemicals which could be future miracle drugs. So, as I said, the factors, identification of plant resources is important. So are also important the factors that influence those plant sources. So, when we start with phytochemical research, we do certain level of extractions and all of that, but there should be a certain difference between the home research and real-time research. So, in a home research, we really don't do any kind of documentation or any kind of documentation is required. Just go ahead, extract, and use for your different purposes. However, when you go for on a scientific platform, you need to have your documentations absolutely clear. So, from the documentation purpose, first and foremost is the taxonomical feature, taxonomical identification, which essentially forms the foundation of the phytochemical research. Apart from the taxonomical identification, it also becomes immensely important to consider some more factors that needs to be documented. Some of them include seasonal variations or geographical location. Different geographical location may have different quality and quantity of phytochemicals, which could be quite crucial in behavior. Even a single change of functional group can immensely change the potential of the phytochemical. So it's immensely important to do a documentation of the geographical location. Second is the plant age. Every plant has its own developmental stages, right? So it is important to capture the plant in the right moment, the right time. It may so happen the specific phytochemical, target phytochemical comes at a certain age after a certain maturity, and then decays start after a certain age factor. For example, what we call the raw, ripe, and the one which is called which gets spoiled, right? So there are three different stages of any kind of plant product decay, right? It starts decaying. So we need to capture certain phytochemicals can be captured when the plant is still in the dormant phase or in the development growing phase. Certain phytochemicals evolve or develop when it reaches its maturity, and certain plant phytochemicals are generated while it starts decaying, right? So it's important to capture the document the right plant age which the plant had been used for isolation. Thereafter, abiotic and biotic conditions are also important. They also influence the plant's growth and development, and is equally is can impact the quality and quantity of the phytochemicals. So to maintain the consistency of these documentation, we use two different techniques, what we call the phytochemical fingerprinting, which is HPLC based, and alternatively, we may use DNA barcoding, which is genetic genome based, right? So we basically use these two different techniques. Either of them can be used to actually observe the consistency of the starting source of the research. That basically gives us a scientific validation of the resource from which I am taking my research from. Thereafter, extraction process, as I said, there is an extraction medium. For example, we all can imagine a utensil. We use certain spices like lavang and turmeric, elaichi, so and so forth. We put in water, boil, take out the extract and drink it, right? So that essentially the water acts as an extraction medium over there. Similarly, depending on the kind of phytochemical that we intend to isolate, we can select a whole range of extraction medium that ranges from extremely polar water to an extremely non-polar hexane, right? Or a different combination of anything between these two extremities, like ethanol, methanol, or even acetone or ethyl acetate, so on and so forth. So all kinds of organic solvents are available, or even an aqueous solvent is available, which we can use for extraction. If it is a polar phytochemical, we use a polar solvent. If it is a non-polar phytochemical that we intend to target, we use a non-polar solvent. So depending on trial and error, we can actually decide the polarity and ultimately decide the extraction medium that we need. That could give us the optimum extraction. Now, apart from the polarity, pH and temperature also play a significant role. The pH of the medium, of extraction medium, has to be optimum. It can be acidic, or it can be neutral, or even it can be basic, right? So depending on the kind of phytochemicals, on the pH of the medium, the phytochemicals can be, it helps the phytochemicals to be extracted and solubilized within the extraction medium, leave the plant part and enter the extraction medium, right? The extraction medium, the phase, there are two phases. One, the phase in which the phytochemical is trapped, and another phase, the extraction medium, in which I want the phytochemical to be liberated in, right? So the pH plays extremely important role in changing the biosolubility of the phytochemicals. So we got to find the optimum pH for really targeting the phytochemicals for getting optimum extraction. Temperature similarly can be altered. Most of the times we use near to boiling temperature so that maximum extraction can take place, because we all know that temperature more or less most of the time increases the solubility of the phytochemical solubility, increases solubility of phytochemicals. So temperature becomes crucial. So fourth and the lastly, the method of extraction. The most common of all is the Soxhlet apparatus, using Soxhlet system of extraction and maceration. Soxhlet apparatus essentially is more like a steam distillation or a simple distillation technique, right? Condensation, distillation, and condensation takes side by side, and essentially it meets the near to boiling temperature of the extraction medium. So it gives a good efficiency of phytochemical liberation. Same is the case with maceration, but the difference between succulent apparatus and maceration is that maceration takes place at the room temperature or even at a cold temperature, whereas succulent apparatus uses high near to boiling temperature. However, the new techniques which are coming up right now are the ultrasonic and the microwaves and subcritical water extraction. These are the latest techniques which are into the research right now, extensively being used in the research as a research tool for extraction method. And the good part about these two techniques are that they are completely solvent free, right? We don't need to use the toxic organic solvents. Whereas the limitation of these techniques is that the yield would be quite low as compared to the maceration and substrate apparatus. So both these two different domains, the old school of substrate apparatus and maceration has a high yield, but the toxicity issue becomes the limiting factor. Whereas the new school technique of ultrasonic waves and subcritical water extraction is a new school technique where toxicity issue is completely ruled out. However, the yield could be a bit compromising. Once the extraction is done, we have a number of compounds within the extract, and maybe hundreds of thousands. All kinds of compounds would be there. So isolation is quite important. For isolation, we use chromatographic techniques. The most common of all is the column chromatography, and the HPLC, and in parallel, we can also use gas chromatography. So gas chromatography essentially is for volatile derivatives, which essentially can withstand high temperature, whereas alternatively, the normal column chromatography is beneficial for those which cannot withstand high temperature. So in either case, we can have a good separation. The limitation of gas chromatography is that the compound which is separated can basically, it's difficult to retain them. Whereas in preparative SPLC, you can always retain and the aliquots of the separated compounds, so that you can go ahead with the further investigation. So once separated, you have different compounds. You can go ahead with biochemical analysis for your compound categorization, whether it is an alkaloid, whether it is a terpenoid, whether it is a flavonoid, or whether it is a polyphenol, so on and so forth. You can use various biochemical assays for both qualitative as well as quantitative phytochemical analysis for documenting that. So compound identification is the ultimate target where we use mass spectroscopy these days. Previously, we did not have mass. As the evolution of techniques have happened, we also have utilized those techniques for our own improvisation. So mass spectroscopy essentially helps us in identification of the isolated compound because of its specific characteristic mass to charge ratio that it has. Most of the time, this mass spectroscopy comes in combination with the liquid chromatography or even gas chromatography, so that as and when it can happen on the run, it can happen. Isolation as well as identification can happen on a single go. Now, suppose if you are lucky enough to really get some kind of unknown compound, which is not detectable by mass spectroscopy or does not have any previous database in its mass spectroscopic database, does not have any reference, we can always go ahead with other spectroscopic techniques like nuclear magnetic resonance, which helps in structural elucidation of the new compounds that we have isolated, and thereby we can actually come up with discover some new compounds which have not previously been reported, and that would be the ultimate thing that any researcher could desire for. So the application potential for these isolated compounds has four major domains. I divided into four major domains. It could be many more, but essentially what I could come up with was these four major domains of medicine, culinary, textiles, and agriculture. Culinary, a number of natural dyes and aromatic flavoring agents. For example, we know saffron, right? Saffron is an extremely costly and rare endemic phytochemical, which comes from a specific plant, the saffron plant, right? The flower, the stamen of the flower basically contains these phytochemicals, the saffron. So it's expensive, right? So, but at the same time, it's natural and it's quite, it doesn't come easy, right? So it acts as a flavoring agent, and at the same time, since we know most of the phytochemicals are very good antioxidants, right? So it can also be used as a natural preservatives for various eatable items. Thereafter, we have textiles. In the textiles, we have all kinds of synthetic dyes these days, because earlier, before the advent of the industrial revolution, we had the source of the dyes were all coming from natural indigo dyes, right? The natural plants. But since they became quite expensive and difficult to manage the quality, we could not pass the quality control. We shifted to synthetic dyes, but the limitation of these synthetic dyes is that these are not eco-friendly. This causes immense damage to our ecosystem. So alternatively, since now we have those modern tools that we can actually maintain the quality control of the natural dyes as well, so which could always be a good option for opting for eco-friendly coloring dyes, which can be isolated from the natural resources. Agriculture, again, we have the latest trend of organic farming, which has been coming back now after the food revolution that we had, the food revolution that we had in the 1970s from Dr. Subramanian. So now we are coming back to the demand of organic farming is constantly increasing, and we also know that the pests and the fungus and the weeds, which can really impact the crops. Organic farming is not cheap; it's expensive. At the same time, we cannot compromise on losing out crops yield, right? So the crop yield should be controlled by using organic pesticides and fungicides, which would be basically coming from most of the phytochemicals, has this potential for acting as a as a pesticide and a fungicide. Now these are basically organic. They could be classified as organic in nature because the ultimate source is the plants, various different kinds of plants. At the same time, they comes from nature and they go into nature. And at the same time, they can also be used for identification of any kind of adulterations of the seeds that are being purchased. The farmers purchase the seeds from the vendors, which could be a fictitious seed, right? Or maybe of a lower variety that the farmer had actually intended to purchase. So that basically, if at all a government could take an initiative and start certification of those seeds and certification of purity of those seeds based on the phytochemical fingerprinting, that could be a really good thing for the farmers to have their fair share of benefit. So currently now in our lab, we are currently working in the phytomedicine field, and we're trying to observe different kinds of phytochemicals and their potential applications in the different medicinal field, right? For example, we have a number of phytochemicals we have and that have pharmaceutical potential as well as nutraceutical potential. So we are trying to give a phytomedicine, which what we qualify as generally the herbal medicines or all kinds of traditional medicine system or the folk medicine system that comes through traditions. We're trying to give them a some level of scientific validation or scientific accreditation, and also at the same time try to streamline some level of quality control for the better reliability. So phytomedicine, though it's a very old, one of the oldest system of medicine, the natural source of medicine has been the plants, and all other new medicine, as I said, has been basically inspired from the phytochemicals that have been isolated from the plants at some point of time. However, the traditional system of medicine doesn't die out. It's still there. We are the different forms of traditional medicine system, the Chinese tradition system of medicine, or the Ayurvedic system of medicine, or the Unani system of medicine. So all of these are basically plant-based, right? So the plant, these medicine basically they're still there with us, and they're still fighting for the deserving place in the in the modern concept. So what we are trying to do in our lab is trying to use these modern tools of phytomedicine and try to advance their reliability and their performance through various techniques such as chromatography and spectroscopy. These are one of the oldest techniques even in modern tools that has actually helped us in identification of a number of phytochemicals. And and that has been in the late 1960s and 70s. Spectroscopy was in use demand. Even now, it's only rising. The demand of spectroscopy is only rising. As I said, NMR is used for structural elucidation. Thereafter, we have molecular biology tools, number of molecular biology tools and cell culture techniques, which essentially helps us in fast screening of various phytochemicals for their potential therapeutic potential, right? So it becomes much easier to prove into in vitro and in vivo models rather than directly going into clinical trials. There is no point of clinical trials in this because this already time-tested medicine. But to have a beginning of the scientific documentation, they're using these techniques of molecular biology and cell culture techniques to start off with some level of scientific validations. Thereafter, since the 1990s, in the last 30 years or so, we have been coming up with new techniques of nanotechnology and computational biology. And nanotechnology have been started in the 1990s, and in the last 2 decades has really caught up with the scientific fraternity. And it has been explored as a potential biomedical tool, right? So we are using the same technique of biomedical tool, nanotechnology as a biomedical tool, and trying to see its purpose in phytomedicine, trying to find its position in the ladder of the phytomedicine, where it can really contribute to phytomedicine. And computational biology is another and very important upcoming tool that actually can help us in in predicting and designing of phytochemicals, new phytochemicals or synthetic phytochemicals, which we'll be discussing in the coming slides. So the concept of the reverse pharmacology in phytomedicine, as I said, the computational biology tool is coming in a big way, is going to play a role in a big way in designing of phytomedicine, medicine in general, and phytomedicine in specific in this context. So reverse pharmacology essentially talks about in silico modeling of drug design and going from in silico going to in vivo and then to the clinical trials. Previously, before the advent of computational biology tools, the scientists used to design and synthesize all kinds of molecules within the lab, and out of those thousands of molecules, two or three could actually enter the in vitro trials, in vitro studies, and in vivo, and then finally clinical trials. So it was quite a tedious job, and it's very expensive. So with the advent of in silico modeling, basically it helps us in trying to design the phytochemicals, for that matter, even any kind of molecule, which could actually help us in prediction whether that particular molecule could have any kind of pharmacological properties or not. One of those very good tools you can go ahead and check out is Swiss ADME. This Swiss ADME is one of those computational biology platforms which can help us in designing and checking the pharmaceutical possibility of that particular novel design molecule, right? So Swiss ADME could be a very good option that you can check out. And in addition to modification of molecules, you can also go out and check out. There's a huge database of phytochemicals, structure of phytochemicals in PubChem, or you can also check out some other tools like Zinc database that basically helps us in screening drug candidates for suitable drug targets, right? Disease targets. So, for example, there are all kinds of diseases present in the all kinds of diseases happening, right? So, and all kinds of drug tests are going on. So, instead of going for directly for in vitro and in vivo, if at all we can go a step back and check out whether there is any, if at all, any possibility of feasibility of in silico prediction. In silico prediction is still quite new, and it still requires validation, its own validation in reverse. But today, if we start, maybe down the line in 10 years, we could make it more efficient in prediction of drug candidates. So in addition to the drug candidates, it also helps us in simulation studies where we can mimic the entire microenvironment of a particular tissue or a cell, where we can simulate a physiological environment where it can actually help us in understanding how the any molecule would actually behave, right, in that particular environment. And basically that would help us in trying to becoming more realistic about replicating the physiological environment in silico modeling. So that all of this is basically quite in a preliminary stage, and we are moving quite fast towards the direction that probably within 10 years we would have we'll come to the point where we have in silico as a reliable platform for starting off any drug discovery and development. So the second concept of nanotechnology in phytomedicine, basically nanotechnology essentially involves synthesis of nanoparticles, nanostructures, which is in nanometer range, and basically they have diverse applications even in medicine. Some of the application that we see that we have very popularly known gold and silver nanoparticles, which are being intensively studied for their anti-cancer potential. The results are quite satisfying, and they're moving in a rapid direction. So it could really come up with an alternative to toxic phytochemical, toxic synthetic medicine. However, nanotechnology, when we apply in phytomedicine, the phytochemicals are, as I told, these are antioxidants. They are reactive in nature, and they basically help us in synthesis of these inorganic nanoparticles, the gold and the silver nanoparticles. What we call the green synthesis. This concept of green synthesis basically helps us in utilizing the plant extracts in synthesis of various types of inorganic nanoparticles which could have a therapeutic potential. Apart from that, nanotechnology, we also have nanoparticles which acts as a more like a drug delivery vehicle, and there are a number of proofs which actually suggest that nanoparticles help in cellular uptake of phytochemicals, those phytochemicals which are otherwise rejected. We know that curcumin, in spite of having a very good therapeutic potential, and the limitation of curcumin is that it has a very poor bioavailability. So nanotechnology in that aspect helps us in enhancing the availability of this particular curcumin, and helps in cellular uptake. Recently, we had published a paper where we had proven how these nanoparticles, the polymeric nanoparticle, the PLG nanoparticle, actually enhance the cellular uptake of curcumin, and thereby increasing its cytotoxicity. So that is it. And another one, the very important, is the blood-brain barrier. So we know that we all are familiar with the limitations of neuro-related drugs. The limitation of those drugs is that it's difficult for them to cross the blood-brain barrier. So we are also actively working the nanotechnology. People who are working in nanotechnology are actively involved in trying to explore this particular technique in helping transport the drug from across the blood-brain barrier through nanoparticles. And there are satisfying results of in vitro and in vivo. And however, the limitation of this nanotechnology in medicine is that there are very limited clinical trials present until date. As you can see the data below. There are only since in between these 30 years from 1991 to 2020, there are only 698 reported clinical trials. That is for the total medicinal application of nanotechnology. If you open up PubMed and use the keywords nanotechnology and medicine, you would get a clinical trial count of 698. Now, imagine if we take up phytomedicine or transport of phytochemicals in clinical trial, that again reduces drastically. But then the good thing is that that opens up the scope for further detailed research for us. So things that are not really explored till date actually opens up venues for us for exploration and coming up with feasible options for enhancing drug behavior. So at Nanobiotechnology and Applied Phytochemistry Lab, we are essentially trying to have taxonomical classification based on phytochemical fingerprinting. We are also optimizing various phytochemical extraction processes. We are also trying to study the mechanism of action of polyherbal concoctions, essentially with the focus on synergism. Synergism is one idea where, in comparison to the modern day drug system of medicine, where at the most we have combination therapy where you use one or two combination of two different drugs and to get a better yield. However, in this case of phytomedicine, we intend to highlight how it has an inherent property of synergistic action, and that basically is another subject of research that scientists are extremely taking up extremely seriously with regards to phytomedicine. So bioavailability, again, as I said, trying to explore nanotechnology with enhancement of bioavailability. And ultimately, yeah, we do use computational biology techniques to modify different phytochemicals so that we could enhance and check whether there is an enhancement of any therapeutic potential. So right now, we are doing the other way around. We are taking different reported phytochemicals or reported drugs and of known cytotoxicity and checking their behavior in silico, so that we could confirm our in silico based on our in vivo results. So, but hopefully down the line in 10 years, we expect it to be the other way around, the in silico actually guiding us into in vivo. And ultimately the loop side of the all this of the phytomedicine is its scientific validation, and we are extensively working on summation of these scientific validations, and one at a time. There's an entire ocean of phytomedicine that needs to be documented or given scientific validation. We are into that, and there are like various other scientists who are into this. Excuse me. So hopefully we would be able to validate one by one most of the phytomedicines, crucial phytomedicines, which could bring them at par with the modern day medicine system. Some of the important diseases that are being studied for phytomedicine can be differentiated between non-infectious and infectious diseases. Within the non-infectious diseases, we have diabetes, cancer, cardiovascular diseases, and a number of neurological and muscular disorders. All kinds of infectious diseases, bacterial, parasitical, fungal, and viral infections have been studied through practice of traditional medicine. All we are trying to do is trying to give them a scientific validation. That is what we are trying to prove. New viral infections like even COVID. I just checked out my cross-checked my database, and I found that already scientific in silico work for COVID has started through a number of phytochemicals showing potential results. Though these are just in silico validations, we require in vitro and in vivo validation as well for confirmation of that. So as of now, we don't have any specific phytochemical which will say that it has certain any anti-COVID potential, but we are into that direction. So diabetes, as I said, one of the stories is of metformin. Metformin was basically is a synthetic molecule, but its resource was never synthetic. It was always a natural source of Gallagher officinalis, which up till now, until the 17th century, it was used as an herbal drug, herbal medicine in the European countries where this plant is actually found in the temperate region. It grows well in the temperate regions. So in the 17th century, it was a very important medicinal plant. It was actually a weed, but we used to use as a medicinal plant. And in the 18th century, 19th century, they actually found that guanidine is the molecule which is actually giving that medicinal property to that particular plant. So guanidine also was observed to have also used to control frequent urination, which is one of the important symptoms of diabetes. So it was for frequent urine that up till that time diabetes was not really established. The concept of diabetes was not there, but then the disease was there, which was whose symptom was frequent urination. They used to consider that as still a disease, and they used to say, okay, you take this particular medicine, this particular extract, and then your frequent urination would be controlled. So the limitation of guanidine was that it was quite toxic. So isolated guanidine could never be used because of its toxicity issues. It could never enter the clinical trials. However, alternatively, we could find Galligine. Galligine in 1920s, when the isolation and extraction process became much more efficient, the scientists came up with Galligine from the same plant, from the same extract. They could identify another molecule which is a derivative of guanidine with an isopentenyl. One second, please. Yeah, isopentenyl group addition of this to guanidine. So Galligine had a good therapeutic potential, but at the same time, its toxicity was also less. However, the therapeutic potential was not as significant as it was of guanidine. So by the late 1930s, they come up with the synthesis of the organic compounds, and in this case, they synthesized biguanides. Biguanides basically was two guanidine was condensation reaction of two guanidines gave us biguanides. So we could see this is one guanidine, and then we have another guanidine, right? So these two guanidines was in condensed, and basically we got guanides, biguanides. So biguanides basically had a good therapeutic potential, as well as the toxicity was quite less as compared to single guanidine molecule. So toxicity-wise, biguanides were excellent. At the same time, it had certain medicinal properties, but the scientists went ahead and they started modifying these particular biguanides, and a number of derivatives were synthesized. So they took up the concept of galegine and biguanide, and they tried to highlight these methyl groups and these two methyl groups, and they tried to put that into biguanides, right? This particular nitrogen. So, which essentially gave us the formation of metformin in the end of the 1949. We got metformin. So metformin basically, you could see the entire part where a biguanide was a combination of a biguanide and a galegine, right? So it had the properties of both. It had less toxicity. It had better medicinal properties. Well, then after that, this particular scientist, Jean Sterin, he was the one who actually took that next leap of taking metformin into the clinical trials, and then where we had these clinical trials taken into diabetes, and ultimately metformin became the standard drug for diabetes. And till date, we have this metformin as the standard drug. So statistics basically shows how the increase in the phytochemical research on phytomedicine is related to phytomedicine, they increasing each year on year. For this particular graph, we have selected curcumin and resveratrol and digoxin, one of the most extensively studied phytochemicals. So essentially, this gives us a hope that we are moving into the right direction, and someday we would come up with another digoxin or another quinine, which could be helpful for mankind. So important takeaways from my today's talk is that phytochemistry essentially encompasses a wide range of applications, and researchers from diverse disciplines can actually contribute in the advancement of the subject. No specific researcher can really single-handedly help take ahead the applied phytochemistry subject single-handedly. We need to hold our hands and work in collaboration with diverse disciplines and come up what is best for the mankind. So maybe you could be the next part of the next metformin story, like how metformin started from one single plant in the 17th century and ended up with the miracle drug of metformin. As I said, 80% of the plants are yet to be explored, and who knows, tomorrow we could be part of that metformin story, right? So that is the optimism that we need to carry forward. And this basically help us unlock a vast reserve of unexplored flora for the well-being of mankind. So thank you. With that, I conclude my slide presentation, and you all are welcome for suggestions and criticism.
Okay. Thank you, Dr. Hoda, for trying to give us some different aspects about phytochemistry and its applications in basic. I'll take up some questions. Shall we start with the questions, Dr. Alim, or you want to say something?
Yeah. Proceed with it.
Okay. So, Dr. Hoda, I'll take up the questions which have been over there. So, one
Yes, please.
question is from Mr. Sudeep, who is asking, how can you check the biocompatibility of a particular bioactive compound?
Sorry?
How can you check the biocompatibility of a particular bioactive compound?
So biocompatibility essentially means I think it's echoing. I don't know why it's echoing.
Dr. Alim, just switch off your microphone. Yeah, even I.
Yeah, okay, that's good. So biocompatibility depends on its applications. For example, I said if it is a textile-based application, you don't need to worry about biocompatibility because anyway, it's from the natural natural source, it goes into nature. It comes from nature, it goes into nature. So we don't really need to worry about biocompatible. However, when it comes to culinary or agriculture or medicine application, biocompatibility becomes essential, and that comes from the trial and error. We got to test every phytochemical, any chemical for that matter, any molecule for that matter, has to go through biocompatibility tests through various various parameters to really give us an insight whether it's feasible for example, we know curcumin. Curcumin, though in nutraceutical potential, it has nutraceutical potential because of its limited biocompatibility, bioavailability. It cannot really go into therapeutic potential because of limited concentration, but it can always be used as a nutraceutical compound. So biocompatibility essentially comes with the test. You do various tests of biocompatibility depending on our application. I hope that answers the question.
Yeah, there's a second question, Dr. Hoda, who is by Mr. Sudeep only. He's asking how is the bioactive compound, how does it inhibit biofilm formation?
Biocompound inhibits, it's basically biofilm essentially. Can you just mute? Yeah. Biofilm essentially is a microbial layer that basically gets generated over an exposed area of a skin or something, or a wound. So any phytochemical which has an antimicrobial property would inhibit the multiplication of those microbes on the surface of the wound. So that way it can simply prevent biofilm formation. So it's simple work of antimicrobial property. Any phytochemical that has antimicrobial property could prevent biofilm formation.
Okay, that's what they're asking us, and how does it do that? So can you comment on that?
Well, there's an entire mechanism of working behind it. That goes to molecular level. How we need to inhibit microbial multiplication, right? It inhibits different phytochemicals work in a different mechanism. Various mechanisms are there, and as I said, phytochemicals as of now not really been explored on molecular mechanism of action for various applications. So, and especially in case of diseases. So we are still working on that. Since my field of work is not related to antimicrobials, so probably I will not be able to answer the exact molecular mechanism of action. However, there is an entire mechanism where either it could inhibit the protein synthesis, or it could inhibit the transcription, or even or even the replication aspect of the microbe multiplication. So that way they could prevent the biofilm formation.
Okay, there's another question which has been asked by Miss Serena, and she wants to know, is there any negative impact of phytochemicals on human health?
We are still to see every phytochemical has its own pros and cons. It's not that if it's a synthetic molecule, it will not have any kind of. It will have only. It will have all kinds of negative impact on the body. There's a balance between negative and the positive impact. Same is the case with any kind of phytochemicals. It can have a positive as well as a negative. If taken in low concentration, it can be good. It can, if it's taken overdose, it can cause bad impact. So any phytochemicals can have negative and positive for that matter. Let's say, for example, let's say digoxin is a very from digitalis purpurea. You know, we all have heard about toxic plants or poisonous plants, right? So these are poisonous plants. Essentially, what poisonous? It's poisonous because of specific phytochemicals present in that, right? So that specific phytochemicals can be toxic. So it's poison. It can also act as a poison. It can also act as a therapeutic molecule. So it depends. You have to be very careful. That's where the science comes in to really check whether it has any therapeutic potential or not. For that matter, even snake poison has snake venom is a poison, right? But it does have therapeutic potential. Same is the case with plant phytochemicals. It also can be toxic as well. So you have to be very careful. You cannot simply take any phytochemical and say it's okay, it's completely safe. In fact, we had a recent case. You know, curcumin. Some guy in US he injected curcumin, assuming that it's completely safe, and he actually injected curcumin directly into his veins, and he died of toxicity because overdose of curcumin. It's harmful, right? So there's a way of taking in curcumin, that is through oral means. We take curcumin through oral means. We do not inject it, right? When it goes into the stomach, it gets absorbed within the body in the optimal concentrations that the body allows it to be absorbed, right? So that its nutraceutical benefit is experienced by the body. So it's all nature designed system. There is a balance between at all levels.
Okay, there's another question in which it's since you discussed about the concept and applications of nanotechnology. So, can you comment on any application wherein nanotechnology or it can be used for neurodegenerative diseases?
Yeah, there are a number of scientists who are working in this direction, and the biggest limitation of neurological diseases was that the drugs which are designed for neurological disorders has to basically they take into considerations whether it can cross blood-brain barrier or not. A number of phytochemicals or not, even any other chemicals, they have good good potential of reversing neurological disorders. However, the limitation is that they're easily rejected by the blood-brain barrier. So you have to explore what is blood-brain barrier. It's the membrane which essentially prevents uh, all kinds of molecules from passing from the bloodstream into the brain. So that basically preserves the brain from any kind of interference. However, currently there are a number of groups, scientific groups, who are working into this direction. They're using nanoparticles as a drug delivery vehicle, where they're loading their drug candidate and trying to see whether it can really cross the blood-brain barrier or not. And there are positive results for that. But as I said, at the clinical trials, very few of them have actually reached the clinical trials for various reasons. And but then that still opens up the scope for investigation for us. So they are already working into that direction.
Okay, the another question, as I said in the previous talk as well, no discussion today can be without COVID. So one question regarding on that as well. So, are you aware of? Basically, it's more of a on which you can comment about if you have any understanding about it. So, are there any in silico or computational biology studies through which some phytochemicals they have been really, identified, which are possible candidates or possibly effective against SARS-CoV-2?
Yes, there are already since as the COVID started, and then we had an extreme surge of research publications within a year. We could see no other disease has been explored in such a short time to that extent. So we already have like in any other scientific group, a number of other phytochemical researchers have explored this domain, and though in vitro in vivo is not possible because of the obvious reasons of COVID limitations. But then in silico work are already being done, and they are showing good results. They are very optimistic about it. But then in silico remains in silico unless we don't go for in vitro and in vivo. So in silico results are already there. You can actually check out on PubMed, right? You can simply enter COVID-19 and enter phytochemicals. You will be getting whole number of lists of research publications which has reported molecular docking results, in silico results on phytochemicals. Actually, I have also published, I am on the way of publishing one of them. It's under review right now. So where I have basically taken thymol and trying to explore how COVID-19 protein, the virus protein, capsid protein which is there, we're trying to inhibit that. So we are also working on in that direction, and then that the paper is still under review, and let's see what is the response of the reviewers. Undergoing that. Thank you.
There's another question which I think is a very interesting one. I think I like this question a lot. Even I am curious about it. So it is about why biomolecules, functionalized nanoparticles, are getting advantage as compared to naked nanoparticles.
Sorry, please come again.
Yeah, I'll repeat it. The question is, why are the biomolecules functionalized nanoparticles? Basically, biomolecules in association with nanoparticles. So, how
Yeah.
are they of an advantage as compared to naked nanoparticles? Because even nanoparticles have a lot of
Okay,
properties and a lot
okay.
of
So this is
advantages.
totally different from the pure nanotech, nano subject, right? So, see, a naked nanoparticle is an old school now. The researcher in this direction is moving extremely fast, and simply a naked nanoparticle is quite old right now, and it can only help in the passive targeting of the drugs, right? However, surface functionalization of the nanoparticles, that surface functionalization can be either of of any protein or a nucleotide or an oligonucleotide or a peptide, or even small molecules, carbohydrate, maybe it may be a lipid, right? So all of these basically are surface functionalized on the nanoparticles surface, and that basically helps us in active targeting of that particular nanoparticle. So the point here is naked nanoparticles can only take it to the extent of passive targeting of the drugs. And by passive targeting, I mean there's no specific fixed target. For example, there are major restriction sites in our body, right? For example, the liver, right? The majority of the nanoparticles that you induce basically gets entrapped either spleen or in the liver, right? So that basically gives us an idea of passive targeting. However, on the other side, if you have surface functionalized nanoparticles, right, that basically can help in active targeting. Now, for example, we have a specific cancer molecule, cancer cell, where we have a specific receptor target which is exclusively expressed on that particular cancer cell, right? So what we do is basically use a counter protein of that, a counter ligand of that particular receptor, and bind it, coat it on the surface of the nanoparticle, and then put it into the system. So what happens? It works like a ligand receptor interaction, and this particular nanoparticle go and binds to specific cancer tissues, which expresses that receptor, right? So essentially, that particular part type of targeting is called active targeting, and right now in the last 5 years, most of the researchers have extensively focused on active targeting rather than just restricting ourselves to passive targeting. Dr. Sada, you are still muted.
So, just one second.
Yeah.
Okay, so there are two more questions, and I think I'll put them in front of both of you together, and you can answer them in a combined manner or separately as you feel comfortable.
Sure.
So the first thing is, is it possible that the temperature degenerates the phytochemical during extraction process? And the second part is there any reverse drug interaction between a phytochemical and a nanoparticle?
Okay, fine. Yeah. So first, the first question was, can you just repeat that, please?
Okay, we'll take it one by one. The first
Take it one
question
by
is
one. Yeah.
the effect of temperature on phytochemical during extraction process.
All right. So yeah, depends on the phytochemical. Some phytochemicals are they do not degenerate because unlike biomacromolecules, we have two kinds of molecules: one small molecules, another biomolecules. Biomolecules you are familiar with, that includes carbohydrates, lipids, proteins, and nucleic acids, right? So those biomolecules essentially are quite temperature sensitive, right? On the contrary, the phytochemicals are not that temperature sensitive, right? Because they are designed by nature because that way, because they are exposed to all kinds of climatic conditions, and depending on the kind of phytochemical we extract. So these are not even enzymes, nor proteins, nor lipids. They are simply small molecules, a chemical molecule, which can withstand extreme conditions. So extraction becomes quite easy, but at the same time, degeneration does take place after a certain point of time. And that is obvious for any chemical for that matter. So we have to be very careful regarding the degeneration aspect of the phytochemical, but most of the time it's not an issue because of the simple fact it's not a biomolecule, but rather it's a small molecule which is more chemically synthesized. I think that answers the question.
Yes. So the last question, which is there for your session, is there any reverse drug reaction between a phytochemical and a nanoparticle?
So yeah, we make sure that when we try to load a specific phytochemical on a specific nanoparticle, or for that matter, any drug for that matter, on trying to load any drug on a phytochemical, we also make sure as a part of our regular research parameters that we characterize, we also make sure that the interaction is permanent and not it's temporary and not permanent, and it and in no way impacts the interaction between the two. The therapeutic potential of the drug does not gets modulated by due to the presence of the nanoparticles in that same microenvironment. So we do those tests as well in the very beginning before even going into in vitro observations, investigations. So we do take that into consideration to make sure that there is no drug-drug interaction or similar drug polymer interaction or drug nanoparticle interaction, or which may negate the therapeutic potential of the drug. We do study that as well.
Thanks a lot, Dr. Hoda, for taking up all the questions in a very pragmatic and a simplified manner. I would now request Dr. Alam to please conclude the scientific session for today, and after that, we'll have vote of thanks with Dr. Mehboob. So, Dr. Alam, please.
Okay. Thank you very much, Dr. Hoda. Dr. Haldar has left the meeting today. So, thank you very much, Dr. Hoda, for your simple and lucid presentation of the whole discipline. We have presented the things so well that it is very helpful for the PG students and scholars. Actually, you have elaborated all the things in so different stages. So thank you very much, and please go ahead with the vote of thanks.
Okay, Dr. Mehboob, please conclude.
Okay, am I audible?
Yes, sir. Please go ahead.
Okay. Thank you so much. I think a very good evening to all. My name is Mehboob Haq, and it is my honor and privilege to propose the vote of thanks for today's session of weekly talk series Bio-Illumination. First and foremost, my heartiest thanks to our Honorable Vice Chancellor, Professor Muhammad Ali sir, who is also the chief patron of this program for his constant guidance and support. I would also like to thank the respected Registrar of Aliah University, Dr. Syed Muddasarul, and Dean, Faculty of Science and Technology, Professor Mehdi Kalam sir, for all the supports and encouragement. Now, I would like to thank our HOD, Dr. Masroor Alam, who is also the chairman of Bio-Illumination 2020, for facilitating the efforts, and his efforts to make this program a success. We also thank you for inviting such an eminent speaker today, Dr. Shubhasis Haldar, and chairing the technical sessions of today. Thank you, Dr. Alam, once again. Now, most importantly, my heartfelt gratitude and thanks to Dr. Shubhasis Haldar, our eminent speaker of the first technical talk of today, for accepting our invitation, and he for agreeing to give a talk despite his busy schedule. Thank you, Dr. Haldar, though he is not here, for his very informative and interesting speech, which is very promising as well in future course of modern biology research. That the single molecule technology, a new window to see complex biological phenomena, which will be very beneficial for the budding researchers for sure. I proudly express my heartfelt gratitude and thanks to our very own Dr. Muddasarul Hoda for his very informative and interesting talk on applied phytochemistry as an interdisciplinary research concept. I am sure all the young researchers will be immensely benefited. My heartfelt thanks to the convener of Bio-Illumination 2020, Dr. Safdar Ali, for smooth moderation of today's program, also for taking all the technical efforts for live streaming of the program in YouTube. Thank you, Dr. Ali, for all your efforts and hard work. My special thanks and gratitude to Professor B.N. Chakraborty for your constant guidance, support, and encouragement at every step of this endeavor. Thank you, sir. Like it is said, success of any program depends on strong collaborative efforts. Here, my special thanks to all my dear colleagues of the Department of Biological Sciences, especially Dr. Muhammad Zakir Hussain, Dr. Sheikh Kabita, and Dr. Sheikh Muhammad Abu Imam Sadi. Thank you all for your technical supports. Now, all dear participants who joined us in Google Meet as well as in YouTube platform, thank you so much for registering in the program and turning up in great number. Your presence and active participations are our motivation. Now, I'd like to take the opportunity to announce that the link of, actually I have seen many of the participants were asking for the feedback. So I would like to mention that the link to the feedback form will be shared by email, upon filling which the e-certificates will be issued. But one important point is that the link will be active till 14th of September. So you have to fill the form within 2 days, or else you will not be issued a certificate. Moreover, I will also share the link to for registration of our next week's talk in the same email. Anyway, thanks once again. Thank you so much for your attention. Now, that's it from my.
Lastly, before ending, just I want to thank from the whole family of the Department of Biosciences to Dr. Mehboob. I think he is taking the maximum pain of just creating Google Forms and just making the communication with so many participants. So thank you very much from Dr. Mehboob for all of your hard work. Thank you very much.
Thank you. I think with the permission of all the dignitaries, we can end the program now.
Yes, sir.
Okay. Yeah. Okay. Then the participants now can leave the meeting.